How does the temperature, pH, and substrate concentration affect enzyme rates? Remember that enzymes are protein molecules that help speed up chemical reactions, and they either do one of two things. They either break things down or join things together. It's also really important to remember that enzymes are reusable, and some enzymes can react with 1,000,000 molecules per second. Let's first talk about substrate concentration. Substrate concentration refers to how much of the substrate is going to be present for the enzyme to react with. And as you can see in our graph, the higher substrate concentration there is, the more reaction is going to take place. This is because there is just more substrate for the enzyme to react with. Remember that enzymes aren't living, so they're not going to seek out and try to find their substrate. Enzymes can only react with their substrate when their active site bumps into that substrate. So if we think about it, the more substrate that is present around the enzyme, the more the enzyme is going to react with that substrate. That's until we reach something called the point of saturation, where it doesn't matter how much more substrate there is, the enzyme is working as fast as it can. Temperature is next. There is a specific range at which certain enzymes will work, and that is different for each enzyme, but all enzymes have an optimal temperature. That is the temperature where they're most efficient. Past this point, you're going to see that the enzyme reaction rate goes straight down. That's because at this point, the enzyme or protein is going to denature, which means it starts to break down. When that protein breaks down or denatures, it loses its structure. And when you lose structure, you lose the function. Even in cold temperatures, there might be some reaction, but in the cold temperatures, the enzymes will not denature. All right, now let's talk about pH. pH is pretty similar to how temperature affects enzymes, where there will be a range where the enzyme functions and there will be an optimal pH. However, there will be a point where the pH becomes too basic or too acidic. For this enzyme, past 10 and below 6, the enzyme is going to denature, which again means that it will start to unfold and lose its structure and lose its function. The difference between pH and temperature is that the protein will denature on both sides of this graph, where in the temperature, it only denatured when it got too hot.